And here we go. So again, I'm very glad to be here to present this work to you today. So initially, I was supposed to talk only about the first topic, and I don't know, a couple of days ago, I decided to add a second study that is in fact really a different topic. And looking at the talk this morning, so you'll see that the second talk is really in the theme of the day. So all of it is unpublished data, so feedback is very welcome. And these two studies will look at auditory cortex from very different perspectives. So the first study is really a sensory processing driven, let's say, and looking at temporal integration in the auditory cortex, whereas again, the second study is looking at the interaction between action and perception in the auditory cortex. But first, things first. So temporal integration and how it depends on the cortical hierarchy and on vigilance levels. This work has been spearheaded by Magdalena Sabat, a former PhD student in the lab, now continued by Hortense Gouillet, who is here today, and all the work has been done in close collaboration with Sam Norman-Inouye, that is just here. So to such a venue, I don't need to expand a lot to convince you that natural sound unfolds over multiple timescales. And as a consequence, the brain needs also to be able to integrate sensory information across multiple timescales to process sounds and parse incoming stimuli. So if you just look at these two spectrograms of speech and music, it's very obvious that speech is much faster than music, and it's also revealed in this temporal modulation spectrum here. So again, the rate of temporal variation is stimulus dependent. But actually, if you zoom in into speech, that is a stimulus that we need to sparse, you see that the constituents of speech are of very different durations. Phonemes, a few tenths of milliseconds, syllables, a few hundreds of milliseconds, and words up to a second. So again, there is this wide range of time scales that the brain needs to deal with. And not only that, but actually if you take, let's say, a given phoneme spoken by different speakers, each speaker is a different color, you see that for the same acoustic structure, there is a variable duration depending on the context and the condition. So the big question is, how does the auditory cortex flexibly integrate across diverse and varied timescales? So how to estimate integration window, the duration of integration? So here I'm showing just an acoustic waveform with a putative integration window of a neural unit, shown with a gamma function. And so we can define the integration window of that unit as how long in the past this neuron is integrating information. In other words, it's the time window over which the stimuli will alter the neural response. It's a simple idea, but there is no method that is directly assessing the integration window this way. You may think that using spectrotemporal receptive fields, so STIRF, that has been an extremely powerful approach over the last few decades, we may be able to extract integration window. So STIRF are assessing a linear mapping between a stimulus spectrogram and a neural response, and maybe from the shape of the STIRF, we could extract the duration of integration. Nevertheless, for all of you who've been trying to use this approach, you know that many neurons, especially non-primary auditory cortex, will not show a decent STIRF, so you cannot actually use this approach. Also, the limitation of STIRF is that it's relying on an assumption of linearity, therefore it's missing all the non-linearities in the cortical responses that could, for instance, explain flexible integration. So for this reason, because maybe the STIRF approach is not enough, Sam developed a few years ago a new paradigm called temporal context invariance paradigm, explained a few days ago already, that is without, in an assumption-free manner, making us able to extract the duration of integration of a neural unit. I'm going to try to give you the concept in a couple of slides. So imagine again we have this neural unit that we present with a stream of short sounds. All the short sounds are music, speech, conspecific vocalizations. And you see that in these two different contexts or orders, the unit is integrating these blue segments, but because the surrounding context is different, the response of that unit to the two contexts will be different, just because, again, the surrounding, the preceding context to the blue segment is different. So different context, different response, despite the fact that the blue segment is embedded in the integration window. Now, if we present that neurons with longer segments, you can see that the integration window of that neurons at some point becomes contained within the segment duration. Therefore, at the end of this segment, the neuron will have a response that becomes independent of the surrounding context. It will be a context invariant response. And then we can define the duration of integration as how long it takes that neurons to have a response that becomes independent of the preceding context. Now imagine we have a neuron with a shorter integration window. You can see that even in the condition with very short snippets, it will achieve context invariance because even with short sounds, the integration window of that neuron is contained within the short segments. So again, the way we operationalize our definition of integration window is the minimal duration that is required to achieve a context invariant response. In other words, if I have an integration window of 100 milliseconds, I will achieve a context invariant response for all sounds that are at least 100 milliseconds long. Concretely, how do we compute that? So what we do is that we take all of these segments we have that we reorganize, so they are shown with this gray area, and we're gonna first compute the correlation of the response of the neurons within the same context to all the short segments, and this gives us the same context correlation that we call actually noise ceiling, that gives you a measure at each point in time about how reproducible the response of that neuron is. But then we can also, for each time lag within this segment, compute a correlation between contexts. So when you enter that segment, the response of the neuron is independent across the two contexts because it is integrating the context, the preceding context, that is different in the two orders. But as you correlate time points that are more and more within the shared segment, you see that the correlation is going to increase because the integration of window of the neuron is integrating more and more of the shared segment until at some point it reaches the noise ceiling. And when it reaches this context invariant response is how we define the width of temporal integration window that we can extract with a model, and I will not get in the detail today. So I will call in the future slide this curve an invariance curve, and the key message you have to remember is that the later this blue curve touches the noise ceiling, the longer the integration window. Using that method with macro electro recordings in the human auditory cortex, shown here with a white line, Sam has been showing that there is a hierarchy of temporal integration in the auditory cortex, going from a few tens to 100 milliseconds in primary regions to a few hundred of milliseconds in the non-primary regions of the auditory cortex. So first question coming to mind when you see that is whether this is specific to humans because of the unique demands of processing speech and music, or maybe because it's a feature that is shared across all mammals that have primary and non-primary regions of auditory cortex. Second question we'll ask is whether this integration is flexible with information rate. If I'm exposed to a fast speaker, do I have a shorter window? If I'm exposed to a slow speaker, do I have a longer integration window? And third question we'll try to address is that in the recent years, it has been shown, for example, here by the lab of Stephen David, that the response of a given neuron is largely dependent to a sound, is largely dependent on the pupil size. So we'll try to ask whether the temporal integration vary with fluctuations in internal states. But first, let's go back to the hierarchical organization. So for addressing this question, we turn to the ferret. That is a model that is convenient for this question because it has a very clear hierarchy in the auditory cortex of primary, secondary, and tertiary auditory cortex. We perform single and multi-unit recordings in the primary and non-primary regions of these animals using microelectrode arrays chronically implanted in the brain of these animals, and all the recordings were done head-fixed in passively listening animals, awake. Here, to start with, I'm going to show you two example units, one in the primary auditory cortex, one in the non-primary auditory cortex. Each of these panels corresponds to sounds of different durations. So you see that the gray shaded area is increasing more and more. First, we computed the noise ceiling, if you remember, so we just correlating the response across the same context, same sounds. And then I'm showing you the so-called invariance curve that you see for the primary auditory cortex unit is quickly rising and touching the noise ceiling in about 20 milliseconds. So the integration width of that neuron is pretty short. On the contrary, if we look at a unit from the non-primary auditory cortex, you see that it takes much longer to the invariance curve to reach the noise ceiling, up to 100 milliseconds. So we have a variety of integration already if we look at these two units. Now what we can do is that we can take all units we've been recording in primary and non-primary regions of the auditory cortex and just project them on this schematic view of the auditory cortex with a color code that is going to show you the width, the duration of integration for all these units. And what you see very clearly is that in the ferret, like in human, we have a hierarchical organization of the temporal integration, going from short to longer integration. This spans 3 octaves from 15 milliseconds to more than 125 milliseconds, and with a clear shift of integration width from primary to non-primary regions. So here what we did is that we took all units recording and we actually average all their invariance curves, depending on whether they are close to the primary auditory cortex, top row, or further, the furthest from the auditory cortex, bottom row. And you see again that the closer you are to the auditory cortex, the fastest you reach the noise ceiling. But also you see that virtually all units reach the noise ceiling for segment duration longer than 100 milliseconds, showing that all recording units had a time-limited compact integration window. So now that we revealed this hierarchical organization of integration in the ferret brain, ferret auditory cortex, what about the flexibility of this integration? So for that, what we did was simply to speed up or slow down the sounds we used for estimating the temporal integration. And so we had basically two hypotheses in mind. So here I'm showing you acoustic waveform or compressed or stretched stimuli that we'll use for computing the integration window. And we wanted to test whether, again, the auditory cortex will adapt its integration to the rate of the incoming stimuli. First hypothesis is that, in fact, the integration window will remain fixed independently of the speed of the sound. This is a scenario we call the time-yoked integration because it doesn't vary with the rate of the stimuli. On the contrary, if the auditory cortex is sensitive to the rate of the incoming stimuli, or maybe looking for specific acoustic structure, you would see the integration window of the neurons to compress or stretch with the stimuli. So here, to test for that, we repeated the experiment with compressed, original, and stretched stimuli, and I'm showing you the invariance curve average across all recording neurons, and basically you cannot see any difference between the three conditions, showing that the integration windows are largely invariant to the rate of the incoming stimuli. We can actually report in this scatter plot each individual neurons plotting the integration window duration for the compressed stimuli versus the stretched stimuli. And you see that basically all neurons fall on the green diagonal that corresponds to the time-yoked scenario with the same integration for compressed and stretched stimuli. And we showed that also this is not dependent on the distance to the primary auditory cortex, where the time-yoking scenario turned out to be observed irrespective of the distance to the within cortex. Last, we reasoned that maybe this flexibility couldn't be generic across all sounds, but maybe it could be specific to conspecific vocalizations. So for that, we cluster the sounds into the ferret conspecific vocalizations and human speech. But again, looking at the invariance curves, you see that for the three conditions, they overlap, showing that the integration windows are invariant to the sound rates, but also to the sound category. So just a short interim summary. So I show you that all neurons in the auditory cortex have a time-limited integration window that is hierarchically organized and seems to be invariant to the sound category and to the rate of the sounds. So knowing that, so we looked at whether we can change the integration window with external factors, like the speed of the stimuli, but what about internal factors when the internal state of the animal is fluctuating? So for this, what we did was to replicate the experiment with the same sounds, but now monitoring not only the pupil size, but actually other proxies of arousal in these animals, like the face motion, we heard about it yesterday, but also ECG or the breathing rate of the animal. The idea being that maybe there is not a single dimension of arousal measured by pupil, but maybe there are multiple dimensions of arousal that could be captured by different bodily variables. And indeed, you're going to see it's what we found. So here what I'm showing you is the firing rate of the neurons as a function of the state of each of these bodily variables. So first things first, we find that, as expected from the literature, the wider the pupil of the animal, the higher the firing rate. But then when we looked at other bodily variables, we found that we had other types of relationships, not necessarily monotonic increasing, but even non-monotonic relationships with respect to the heart rate or the breathing rate, for instance, suggesting that maybe there is not only one type of arousal. Now what I'm going to do is that I'm going to swap this y-axis for the duration of integration window to see whether the duration of integration depends on the internal state of the animal. And what we found is that, no, despite the fact that the firing rate is changing a lot with the state of the animal, the integration width seems to be very fixed and stable. So at this point, we are a bit wondering whether we could have this, if only anatomy, position in the hierarchy, is driving the integration. And we thought that maybe we should look at this integration outside of the normal operating regime of the system, and that is why we turn to sleep, to compare wake and sleep. It turns out that in the lab, we have Arseny, shown here, that has been adapting a method used in the mouse to be able to score a sleeping period just based on the olfactory bulb activity, basically the gamma oscillation in the bulb. So when you implant the animal, you end up with a ferret like that, with electrode in the bulb, electrode also over auditory cortex, and even here, this is a neuropixel in a hippocampus. And then this time we are doing experiment freely moving, letting the animal sleep and being awake, and while we play sounds to the animal. So our sleep scoring method is pretty efficient. So this is just a spectrogram of the gamma oscillations in the bulb as the animal is awake and falling asleep. And you see a collapse of gamma oscillations. So now we are able to separate wake and sleep period. And here I'm just showing you one example unit, again, the invariance curve of that unit. And you see that the sleep curve is reaching the noise ceiling faster, suggesting that shorter integration window was observed in sleep compared to wakefulness state. This is kind of consistent with the idea that sleep induces shorter integration window with less sensitivity to the context. It turns out that this animal also had electrodes in the hippocampus, so therefore we could score REM and non-REM sleep based on the theta power. So again, this is the same, at the top you have the same spectrogram that before, so OB gamma, collapse when the animal is falling asleep. Now here I'm plotting the theta power in the hippocampus, and because ferrets are carnivores, they sleep a lot, they do a lot of REM sleep, and you can see this very nice alternance between non-REM, REM, non-REM, REM, non-REM, REM, and so forth. This is very preliminary, but in the few units we've been looking at, it seems that non-REM sleep is, also has a shorter integration window compared to REM sleep, which seems to suggest that the shorter integration window we observe in sleep are driven by non-REM sleep. But this we need to confirm to be sure. So a short summary. So I show you that the integration seems to be independent of fluctuation in the internal states. I couldn't show you that sedation makes integration longer, but what is for sure is that sleeping makes integration shorter. So with all this said, now I'd like to transition to the second part, if time allows. And now I'm really changing gear to talk about action and perception in auditory cortex. So this is really in the theme of what we've been discussing this morning. The good thing for me is that basically all my introduction has been already spelled out this morning. So there are classical examples where action is modulating perception. This morning we saw like this nice illusion with the saccades. There is also with saccades and also the saccade in the train that we don't see when we are in the train. Also self-produced sounds are usually attenuated, like footsteps or chewing. And so understanding the sensory motor interaction are critical because this is also how we learn to vocalize, to speak, to tune an instrument, and how baby learn to speak. So one of the prominent theory to explain how we learn sensory motor interaction is that there is these two internal models that the brain is learning during sensory motor interaction. One of them is what was described this morning as a predictive suppression, which is that within auditory cortex, there will be a comparison between a prediction coming from motor centers, the so-called corollary discharge or efferent-scopy. And so if this prediction matches the incoming sound, you'll have a suppression of incoming stimuli. Then also you have the inverse model, so maybe a kind of version of the ideomotor theory that is postulating that we can map an auditory target to a motor command. Today I'll talk only about the forward model, maybe another day about the inverse model. And our questions so far are very simple, so we are really interested in the dynamics of learning upon exposure, upon implicit learning of the forward model. So I'll try to show you that we found some neuronal correlates of the sensory motor learning. How we did that was we took inspiration, so we had different constraints. The first constraint is that we wanted to have a device that is forcing a tight coupling between the body and the acoustic production. So to try to have a sensory motor task that is kind of sophisticated, like when we to some extent, learn to speak or tune an instrument. So we took inspiration from this instrument. You may know that it's called a theremin, in which the position of your hand will determine the pitch and the loudness of the sound. It's a musical instrument. Also, we wanted an instrument with an intuitive mapping between the body and the acoustic production, because it could be quite hard to make the frets play the piano. So we wanted to have good ways so that a body movement will result into an acoustic feedback. So for that, we called a new machine called the theremin. And the idea is very simple. We're gonna pair the azimuth position of the head of the animal that is shown here in this plot with an acoustic feedback with the frequency that the animal heard is dependent on the head position. So the simplest is really that I show you a short video of that. Okay? Put it again. Okay, so I think it's pretty clear now. So the latency of the feedback is very short. It's about 6 milliseconds. So I think there is really a feeling of agency when the animal are using the device. And so not only they were exposed to this closed loop condition where the acoustic feedback is tied to what they do, but we alternated between closed loop and open loop condition. Open loop is really a playback condition where we play back to the animal what the animal heard in the last 5 minutes. So it's the same acoustic that the animal is hearing, but now the movement of the animal are uncoupled from the sound. And in all the following slides, I will compare the closed loop and the open loop condition to try to see if there is any difference, and if there is, it's that the system is aware or can maybe predict the incoming sound. So the first things we've been doing is very simple, is that we've been looking at classical electrophysiologists to PSTH, so to evoked response, to changes in frequency. Because as you heard, as the animal is moving, we discretize the frequency, so we can define when there is a change in frequency. And as expected, so this is a spiking response of a bunch of units. And you can see an evoked response when there is a change of frequency, which is good. You can also see that in the closed loop condition in red, there is a suppression of evoked response, which is consistent with everything that has been discussing this morning, and also the work of Schneider and Colling in the mouse. So I will not expand on that. So it seems there is some maybe predictive suppression. But also we notice that there is this big increase in sustained activity when you go from open loop to closed loop condition. And this is shown here, so significant. It took us a while to understand really what is going on in this shift of sustained activity. But so long story short, what happens is that there is a motor signal that is present in the auditory cortex of this animal. So here what I'm showing you is again the same PSTHs, evoked responses, spiking activity, time around the change in frequency for the closed loop and the open loop condition. And now we group all the frequency changes as a function of the head speed of the animal, from low to fast. First thing you see is that in both conditions, it seems there is a tuning to the head speed because you see a gradient here of response with the head speed. But definitely the gain of that tuning is much higher in the closed loop compared to the open loop condition. So upon sensory motor coupling, there is a stronger influence of the head speed of the animal onto auditory cortex activity. A motor signal is present there and is dependent on the engagement in the coupling. Then we started to investigate the sensory representation of the frequencies the animal is hearing. So what we did for that was to turn to population level representation. We took all the neurons we had, all the PSTHs also we use as samplings, and we are plotting now here in this 3D PCA the average response to each of the frequencies that the animal could hear, color-coded, in closed loop versus open loop condition. So what is visible maybe in this PCI space is that in the closed loop condition you seem to have a cleaner representation of the frequency axis. And actually, so we quantified this here. So if you look at the pairwise distance between all the frequency pairs, you see that this is higher in the closed loop versus the open loop condition, suggesting an enhanced representation of sounds in A1 during the coupling. So these results I've been showing you are really obtained by combining, averaging across all the sessions we've been exposing the animal to. But there was no, so it seems the auditory cortex has been learning the contingency but there is no dynamics of learning. So recently, we've been implanting animals with arrays of electrodes, both in premotor cortex, that is an input to auditory cortex, and also in A1. And what we've been doing was to now look at the noise correlation between pairs of simultaneously recording neurons in PMC and in A1. And so basically, we are just looking at the trial-to-trial variability between these two neurons and whether it co-fluctuates in time. So we had two results. The first one is that if you compare closed-loop condition to open-loop conditions, you see that the noise correlation is always higher in the closed-loop condition compared to the open-loop condition, suggesting better communication or a common drive between these two areas specific to the closed-loop condition. But more interestingly, that now if we you group all the closed-loop sessions into early, middle, and late exposure over days, you see an increase of this noise correlation over day, suggesting that as time passes by, as the animals have been more and more exposed to the contingency, there is an increase, whatever it is, correlation between PMC and A1. Maybe it's a common input to both areas, maybe it's really like a communication between both. This we don't know yet. So just to summarize, I've been showing you in this second part that there is an implicit learning of the sensory motor contingency, leading to suppression of the responses, but there's also an emergence of a motor signal into auditory cortex and an expansion of the frequency representation. So all this said, I wanted to thank all the team members for the amazing work they do, and obviously also the small participants of the experiments.
Questions? Yeah. Hey, Patrick.
I'm going to ask a Jonathan question. So can you have two ferrets make music together?
No, I don't think so. So next level will be, on the contrary, to train ferrets to produce an acoustic target. So maybe music, but to give them a sequence or at least one tone, and to see whether they can map an acoustic target to the proper motor action. So this is, if you remember, I had my forward and reverse model. So this is how we want to tackle the reverse model approach.
That was really interesting. So for the second part, am I right in that the ferret is not rewarded for those movements that it's making?
No. I mean, it's not rewarded for specific movement. Nevertheless, we motivate the animal to move by adding like a nutritive gel in front of the animal. So the animal is moving because it's exploring just to get a reward. But it's not conditioned to a specific movement or position. We just encourage the animal to move.
Okay, and so I was just wondering whether or not you think reward then modulates. So if you then have trials where the reward is not there, right, does it still move its head after it's learned something? And how does that modulate the firing?
That would be maybe a simpler experiment. So what you are wondering is whether when the animal is disappointing, whether it's the speed signal would go away, for instance? Yeah, so then it would mean that it's not only the sensory motor contingency, but it's more like what the animal is expecting for a reward.
Yes, and to what extent is there both a sensory motor coupling and a sort of a task-like signal? Yeah.
So even in the open loop condition, we still motivate the animal because we want the animal to move also in the open loop. So therefore, it can't be just the reward, right? So it's really dependent on the coupling.
Okay.
That's a good point.
All right. Is it working?
I can hear you, yeah.
Cool. Hey, good talk. So I'm going to talk about I'm going to ask about the first part of the talk. So the difference in integration window size, like between primary and secondary cortex, was like super convincing looking. I also noticed that you had like electrode arrays that were going from like secondary to tertiary and stuff like that. Is there any sign of anything in particular there? Is it just the data is more confusing when you go from...
Yeah, no, as you know, so when we implant non-primary, we are far enough from primary to make sure it's non-primary, but then we have too little in tertiary, at least at the time when we are doing the implantation. So if we have some electrodes in tertiary, the sampling size is very small. So
okay. So just very few in tertiary.
Yeah, but we would expect maybe an increase, but yeah, we don't have convincing data for that.
Okay, thanks.
Thanks for the talk. I'm over here. I was wondering if the temporal integration window had anything to do with alpha oscillations in the brain.
So when the animal is in non-REM sleep, it's known as a slow wave sleep, and we get a shorter integration window. So maybe, so we haven't, I don't really know how to handle that, but maybe this shortening of the integration window is that because there is these prominent oscillations. I know they are not alpha, but just to say that it's, maybe related. So we don't observe alpha, we don't observe alpha in the auditory cortex, at least not to my knowledge. Often we see theta. Theta is all over the place in the ferret, not only in fact or even in hippocampus, but even in cortex we find it sometimes. We haven't chopped the data. We could do that. We could chop the data into periods where we have some type of oscillation and when we don't. But maybe the sleep is the most convincing. So maybe indeed when it's oscillating, it's shrinking the integration window.
Hi. Thanks for this talk. Very interesting. Trying to think of the results from the second half, sort of in the context of David Schneider's work and the tone pips that he would deliver in coordination with either running speed or reaching motion. It seems like there's a crucial difference where the auditory feedback in that context is happening during movement execution rather than during these sort of sustained movements, where in this case the parrot is moving its head and then keeping it there, but still receiving that reafferent kind of feedback. And I guess I'm wondering first of all about the correlations between auditory cortex and PFC, and if you calculate those, say, just after the movement's been executed versus while it's sustaining that movement, and if those correlations different?
So we have had several passes to try to cut the animal movement into movement initiation, and I think it's what you're talking about. So far we've been, so we just need to sit down and try to nail down this problem, but it's a bit a pain to really segment the movement. So we have exactly the same idea, having the idea that maybe before movement initiation there's going to be an incoming signal. That would be super cool. But so it's very simple on some movements, but as you know, the movement of the animal is very difficult to have something that is working all the time. So we want to do that. We haven't looked yet also about the delay between PMC and A1. So maybe this is phase 0, lag 0, but maybe at different lag we could have, again, we cannot really talk about causality, but directionality, and this we haven't done it yet. So this is kind of fresh data, but I was excited to show it. And last, about the Schneider work, I think in most of the studies they are not in position to really see well if in the sustained activity you get a modulation by the locomotion of the animal, because they do 2-photon imaging, and to my understanding, in 2-photon imaging you rarely look at the baseline activity. So you look at evoked responses, so again what we see, the sound evoked response, but the baseline is something that you take out by doing a normalization, so it's maybe not easy to see with 2-photon.
Yeah, they also do a lot of, go ahead.
It's fitting that the last question of the last session will be by Jonathan Fritz.
Yves, a beautiful talk. I'm interested in the second part of the talk in relation, to this task that you have. Well, it's actually a correlation, right? It's not at this point in task. The animal moves its head to the right, generates a certain set of sounds, and then turns its head to the left. So this reminds me a little bit of Dmitry Aronov's study and it could be interesting to explore responses not only in frontal cortex but hippocampus. But have you tried reversing it so that the animal gets used to, let's say, a high frequency to the right, a low to the left, and then flip it to see what happens? So obviously this could be used for a really beautiful set of, memory studies, as you were suggesting before. But it could also be interesting in terms of the reversal study. Like, how do you have two different representations? Because the animal could presumably figure out, if I do this and do that. And the other thing is, just in terms of the behavior, you have it moving continuously to the right. It's not going to a fixed position to achieve a particular sound. So it's really having a dynamic change as it moves. So, it might be worth exploring just a simple version of it where it just goes high versus low. Is there a reason for you to design it for this dynamics?
So that's the next step, really. So you can, with Félicie, we are working on it. So first we wanted to have find a signature of that something is happening when you expose the animal for long. And now that we start to have a grip on proper findings, we're gonna look at the dynamics of learning, which is, as you said, swap the mapping, and then see whether the first mapping is stored, if we reverse again, or whether the second mapping erases the first one. So all these questions are open. So the setup is quite, so it's a piece of engineering, so there is sometimes, we need to move, to have the scientific questions to move with the setup, but so this we're gonna do next, the reversal. Or translation also, but yeah, I think we'll start with reversal.
I think we should stop.